series. It's a great pleasure to welcome you all, and a special welcome to Dr. Steve McConnell, who very kindly has come to talk to us today. So, by way of introduction, I'd like to share a little anecdote. About a decade ago, a whole new class of genetic variation was discovered, called copy number variants. These large-scale deletions and duplications, which amazingly sit in all of our genomes. But these things were fiendishly difficult to find. They literally were the noise in another type of genetic data. And for a while, it was like the Wild West. Everyone was scrabbling to try and understand where these were, what they did. And in the middle of all this, it was just a complete mess. These different data sets didn't overlap at all. You saw some crazy things like entire chromosomes missing in a patient who was otherwise healthy. And in the midst of all this, the McConnell paper came out, and it was like a breath of fresh air, a moment of clarity, of light in the darkness. Suddenly, it made sense, it fitted with the data people were seeing in front of them. And I think, to me, this really captures the, what I'm going to call the signature McConnell move. Starting off with something extremely complicated, doing an amazing job, an elegant job of making it understandable, and bringing clarity to the field. Now, where would somebody learn such incredible skills? I'm pleased to say it was right here in this building where Steve trained as a PhD with Corey Bergman, and that was back in 2004, starting there. After that, he moved to the Broad in Boston, working with David Altshuler and Mark Daly, which is an absolutely impeccable genomic heritage, and of course was then kept there, where he is now the Dorothy and Milton Flyer Professor and the Director of Genetics at the Stanley Center for Psychiatry Research. Now, in the midst of this, his lab covers a dizzying array of things, all of them done with that signature McConnell move. Particularly, there's the single-cell sequencing with RNA, with DropSeq, where his work went from looking at hundreds of cells to tens of thousands, a real amazing 2 orders of magnitude change. Understanding the C4 locus and schizophrenia, one of the most complicated bits of the genome. I think most other people looked at that and just said, I'm just going to remove that from my data. Steve went both feet in and made sense of it, and actually found some very important stuff about microglia and synapse. And then also understanding cancer, the importance of replication timing. And finally, on top of all of this, also software development, particularly genome strip, which is very, very frequently used. I think it's probably one of the most frequently used callers of copy number variation. And so, with that incredible record, it is a delight to welcome him here today to talk about schizophrenia and inherited variation. And please join me in giving him a warm welcome.
Well, what a lovely introduction by a lovely colleague. Thank you. I have to say, I've never so strongly wished that a seminar I was giving was being recorded as this one, because I would, I just really appreciate that stuff, and that was really nice. So as Stefan said, I was a PhD student here. For 8 years, eight long years, 8 long years in a lovely place and an exciting lab. I attended hundreds of seminars in this room, although almost none at this hour of the morning. And you know, it's funny about having been in graduate school for 8 years, because I have a colleague, I mean, I have actually many colleagues now who are studying the genetics of cognitive phenotypes. And the cognitive phenotype that's easiest to collect in really large cohorts is just the number of years of education people have. Of course, it's only a rough proxy for cognitive phenotypes, but it's so abundant and easy to collect that there are tons of genetic studies on it. And I always try to poke them by asking, so if it took me 8 years to get my PhD, do I actually have more years of education than someone who got their PhD in 4 years? And I'm not quite sure how they handle it in analysis, but so today, you know, I actually gave a lot of thought to what I wanted to talk about today, and in fact, decided that I wanted to spend a lot of our time today actually on a kind of a sequel or second chapter to this work on complement component 4 in schizophrenia, and it's actually that a lot of this all the new stuff will be a little rough, because it's actually really the first time I've given a talk about it, but I really want to talk about it here in large part to get the feedback from from people here. And I would actually encourage people to just, this is like a nice, it's an hour and a half time we have, so in addition to the discussion at the end, people should just feel free to ask questions along the way. So I'm really interested to get people's feedback on that work in progress, and in particular, there's so much thoughtful, it ends up connecting on some level with these questions about sex-specific protective effects, and there's so much thoughtful work on that here by Stefan and Donna Whirling and others that I just really wanted to present it here. So I'm going to end up talking about that a lot, and if we end up not having time to get to the second thing on the outline here, which are these new experimental systems, that's okay, but I would just dive in. So, audiences of psychiatrists don't need to be reminded of this, but often the rest of the world does, about schizophrenia, which is that schizophrenia is not the same as psychosis. Psychosis is one symptom of schizophrenia, but it's not the underlying illness. There are these earlier and more continuous features of the illness, loss of cognitive capacity, emotional withdrawal. None of the mechanisms for any of these things are known, and it's obviously one of the reasons that one would try to do human genetics to try to find molecular factors that shape these things. Schizophrenia, the biology of schizophrenia has also always struggled because it's not had the strong pathological findings that, for example, in Alzheimer's and Huntington's, where Alzheimer's pathology led to plaques and led to all these factors in plaques, and Huntington's pathology pointed very quickly to death of medium spiny neurons, and there haven't been anywhere near correspondingly strong pathological findings in schizophrenia. It's actually one reason we did the work to try to develop DROP-seq, to try to be able to see molecular profiles in tissue at much higher resolution than you can with microscopy. But there's always been this one pathological finding that seems to have been made many times about reduced dendritic spine numbers in schizophrenia, particularly on specific populations of upper layer pyramidal neurons in association regions of the cerebral cortex. And I will return to that a little bit later. So first I'm just going to go back over what we've found about C4 in schizophrenia already, because it's going to be important then for understanding the sequel. So it had been known for more than a decade that the human genome's strongest common influence on risk of schizophrenia, the kind of largest risk factor at a population level, resides in the major histocompatibility complex locus on chromosome 6. You can see that on the kind of Manhattan plot of schizophrenia genetics. It's sort of the Burj Khalifa Dubai World Tower, sort of looming high above anything in Manhattan. But for a long time, it was considered this kind of mysterious, intractable fine mapping problem in schizophrenia because the associated SNPs are spread across several megabases of the MHC locus. They show these kind of complex long-range linkage disequilibrium that spans hundreds of genes. People realized quickly that it didn't seem to track with any specific HLA allele or even any pair of HLA alleles. And in fact, after the 1,000 Genomes Project came out, people realized really quickly it didn't even track around the LD around any variant in the 1,000 Genomes Project. So it's really unclear kind of what kind of genetic force could be causing this kind of cryptic, complex genetic association signal. And fortunately, phrases like mysterious, intractable association are less off-putting to students. And so a really brave and wonderful graduate student in my lab, Ashwin Sekar, worked on this problem it actually became his whole PhD thesis. So Ashwin noticed that if you actually looked sort of cohort by cohort, so the idea here was that sort of this complex association signal might arise from multiple things, but they might have different allele frequencies in different parts of the world. So if you looked sort of cohort by cohort, you might get hints from places where there was more of a pointer to a specific place. And he found that in many of these cohorts, there was a stronger signal in this specific part of the MHC locus. It actually turns out we think this has less to do with these being Northern European cohorts than to do with potentially the way in which they were ascertained. These are some of the cohorts with some of the most severe cases. But in these cohorts, there was sort of an association peak in this part of the MHC locus. And even here, though, it was a complex signal that didn't correspond to the linkage disequilibrium around any known variant. But this happened to be a specific part of the MHC locus that we had been thinking about because it contained a very complex form of genome variation, where even, and now this is going back several years ago to where fewer people had been sequenced, but there had been this study that exploited consanguinity in people who were homozygous across the MHC locus to basically completely sequence and assemble the MHC locus in several people. And already in those people, you could see there were several different forms of this locus. Now, this locus has the complement component 4 gene, or actually genes, because there are two different C4 genes, C4A and C4B. And that's a primate-specific specialization. So mice have one C4 gene, but primates have a C4A and a C4B. And C4A and C4B are, so C4 encodes a protein that opsonizes or binds to sites in tissues and then acts as a kind of an eat-me signal. It's like a molecular post-it note that invites phagocytosis by macrophages and it turns out microglia and other phagocytic cells. But in primates, there's two versions of the C4 gene, C4A and C4B. They're almost identical, and yet they have 4 amino acid differences. And those 4 amino acid differences are all right next to each other in the same domain. And in fact, that's the part of the protein that binds to molecular targets in tissues and determines what molecular targets are bound. And so although it's not really as well worked out as it could be, the evidence is that C4A and C4B bind to different molecular targets in tissues due to this protein coding difference in the kind of business part of the protein that's actually been maintained across years of evolution, across all of these primate species, despite the fact that gene conversion keeps homogenizing the rest of the C4 locus. There's also this a very long time ago, a retrovirus hopped into one of the introns of a C4 gene. And so you see in humans both a long version of the C4 gene and a short version that are distinguished by whether or not they have this longer intron. It doesn't change the protein coding sequence, but we've found, and I'll show you in a couple of slides, it actually shapes the regulation of the gene. And it's been named the C4 HERV, or human endogenous retrovirus, because it was a retroviral insertion, although it turns out we see the sequence at the same place in other primate genomes too, so this actually happened a long time ago. And so it's probably been misnamed as a HERV. It probably should have been called a PERV, but that name was already taken. So we will just go with HERV. And actually, so the function of that is actually not known. So we thought, if already in a few sequenced genomes, in like 6 sequenced genomes, there were 4 very different forms of the C4 locus in which people had different copy numbers of C4A, different copy numbers of C4B, long versions, short versions. We thought, well, what if you could actually look in what are all the alleles, right? And how do they shape human phenotypes? And could they trick genome-wide association studies by kind of causing associations to SNPs in complex ways that were hard to get to the bottom of? So Ashwin developed this molecular strategy for identifying all the different alleles of C4. And the idea was he developed molecular assays to detect each of these different C4 gene types, the long A form, the short A form, the long B form, the short B form, and then did these assays by limiting dilution of genomic DNA across thousands of nanoliter droplets in order to get a precise digital count of the number of copies of each C4 gene type in each person's genome. And it's actually it was right about this time that Evan McConnell came into the lab and saw what Ashwin was doing using droplets to do digital counting molecular biology. And that was kind of what within weeks, he started playing with the idea of getting cells into droplets, and so that we could analyze, parallelize the analysis of single-cell gene expression. And so it was really these experiments that led in a kind of a serendipitous way to drop-seq and sort of droplet-based single-cell RNA sequencing. But that's a different story that I'm not telling today. So these are measurements in diploid genomes, but it kind of came up with tricks where by doing these assays in families, we could resolve these measurements into the contribution of the transmitted and untransmitted chromosomes in each person so that we could figure out the chromosome-specific copy number of each of these kinds of C4 genes. And what that let us do was to kind of reconstruct all of the different alleles that we're segregating in these families. And it turns out, actually, by now we've found dozens of different structural forms of the C4 locus. There are these 4 common forms that together make up about 90% of the haplotypes that you see in populations with European ancestry. And there's this long tail of rare and low frequency forms that collectively make up the other 10%. And we see these 4 common ones in every cohort and every population that we look at, but we see different rare ones in different cohorts and different populations. So collectively, we've seen 50 or 60 different forms of this locus. But I'll focus in a lot of the genetic analysis on these 4 most common ones just because we see them so many times that it becomes possible to just relentlessly measure their relationship to schizophrenia and other phenotypes. And so I'll kind of focus on those in these results slides. So it's obviously kind of an exuberant form of variation to be running around our genomes, and we naturally wondered, well, what is just even the first order molecular consequence of having variation like this? That is to say, how does variation like this shape the expression levels of the C4A and C4B genes in the brain? And so we went at this by, we obtained hundreds of post-mortem brain samples, various regions of the brain, hundreds of donors, but basically saw the following pattern in every brain region that we looked at, which is, first of all, the more copies of C4A you have in your genome, the more expression of C4A you have in your brain. The more copies of C4B in your genome, the more expression of C4B in your brain. And those are both kind of fairly intuitive results. Didn't have to be the case, but they're fairly intuitive. But then the surprise, I mentioned this endogenous retroviral element whose function was unknown. We actually found that it tends to increase the expression of C4A relative to C4B. And so basically, to make a long story short, there are all these different alleles running around in human populations. Depending on what combination of alleles you inherit from your parents, you might have either a low level of C4A expression in your brain or a high level or anything in between. But with these results from the postmortem brain tissue, we were able to develop genetic predictors of the level of C4A and C4B expression in the brain as a function of what C4 genotype you have. But of course, it was SNPs that had led us to the MHC locus, and so, at least in schizophrenia. And so it became important to understand how does all of this C4 allelic variation relate to SNPs and SNP haplotypes, the kinds of things that would give a signal in genome-wide association studies. And this was one of the, you always remember in these kind of, everyone kind of knows this feeling. You have these sort of years-long projects where you're working closely with a student or a postdoc, and yet there are these iconic moments where someone comes to you with a figure, and you just kind of never forget how that figure looked like, because that was sort of an aha moment, right? And this was definitely one of those figures where Ashwin just took each of these C4 structures and asked, what SNP haplotypes does he see it on in these families, right? And here, each of these SNP haplotypes is just kind of represented as a barcode here, where one shade of gray is the major allele and one shade of gray is the minor allele. But the really amazing thing was that some of these common, and then each haplotype or person is a really thin row, but they're organized into blocks because you can see there's a pattern, right? So this C4BS allele we pretty much always see on the same SNP haplotype, no matter who we see it in. But this ALBS allele we see on several different SNP haplotypes. They're quite different from one another. And that actually suggests that this form of C4 arose independently several times on several different haplotype backgrounds. And then same thing with ALBL, it's on several haplotype backgrounds, and the same thing with ALAL. And so this actually, within the course of that conversation, we realized two things. First of all, if variation at C4 were affecting a phenotype, it could cause association to nearby SNPs, but it would generate really unconventional patterns of association to nearby SNPs, because no one SNP would be a good proxy for it, and yet it's still not completely random, and so lots of SNPs would be partially correlated with this allelic variation. And so we realized that might, it was precisely the kind of unconventional and complex nature of this association signal that had made it so hard to figure out. And so first insight was this might potentially be able to generate a pattern like that. But then secondly, we realized that it might be possible to analyze these C4 structures by imputation from existing SNP data. And that turned out to be really important, because although Ashwin had developed these molecular assays, the real opportunity was that their SNP data had been collected from tens of thousands of people around the world, and in 30 countries. And it would have been hard to sort of organize a huge molecular study, global, of that. But we were able to do the following very quickly. So the idea was just, could you take someone's SNP data and then use these kind of SNP barcodes to essentially infer what C4 alleles each person has in their genome. And the reason this works is that we all have lots of common ancestors. And so if you match me and another person for a whole series of, for a genomic segment, we have inherited it identical by descent from a common ancestor. And so if we match people across a long series of SNPs across the MHC locus, we can infer what C4 allele they have with fairly high accuracy. So that allowed Ashwin to do in a few weeks what would have taken years to do at a molecular level, which was to analyze C4 variation in almost 30,000 schizophrenia cases and 30-some thousand controls by basically taking the existing SNP data for people, imputing what combination of C4 alleles they had, then using these expression experiments we had done to essentially infer in each person a kind of genetically predicted expression level of C4A and C4B in the brain. And it turned out that the genetic predictor of C4A expression, although not the predictor of C4B expression, defines kind of a local association peak within the MHC locus, and it actually seems to be independent of this other set of variants here. Here, what I've done is I've colored these markers in the MHC by their level of linkage disequilibrium to either this signal over here or to C4, and it just seems to be an independent signal. And in fact, when you do what's called kind of conditional analysis to try to sort of fine map these things, it confirms that these are independent effects, and that when you control for both of them, there's a third effect that sort of emerges out of the shadow. It's a smaller effect that sort of emerges out of the shadow of the other two. That's at this locus called SYNGAP1, which had actually been previously conclusively identified in autism in work by Stefan and others, that loss of function mutations in SYNGAP1 are a fairly high penetrance risk factor for autism. So across this C4 allelic series, schizophrenia risk seems to rise with increasing expression of C4A. That is to say, this is just an analysis of this overall aggregate predictor, but if we go sort of C4 structure by C4 structure and ask does this relationship hold up, it turns out across each of these alleles, the more expression of C4A it drives in the brain, the greater level of schizophrenia risk that it associates with. That was still kind of less conclusive than we wanted. So how could you evaluate C4 as sort of the true cause or driver of this association signal? As you probably noticed, this has been a real challenge in common variant association studies, because sort of everything's in linkage disequilibrium with everything else. So you sort of get all of these ties that you just can't break. But we realized that nature had kind of given us a gift here, precisely because many of these C4 structures had arisen multiple times on different haplotype backgrounds, it made it possible to test the following additional prediction, which is that if C4, rather than other variation in the MHC locus, is the cause of this association signal, then it should be the case that a given C4 allele associates with the same level of schizophrenia risk, regardless of which MHC background it is segregating on. And in fact, that turns out to be the case. So across this allelic series, each of these alleles turns out to have a relatively, each of these C4 structures turns out to have a relatively uniform schizophrenia risk, regardless of the other genetic variation that's segregating with it in the MHC locus. And again, this seems to correspond to the level of C4A expression that's generated by each of these alleles. So, of course, one prediction of this is that if you could actually test brain tissue for C4A expression levels, that it would be elevated in schizophrenia patients relative to in controls. That's not an analysis we can do in 30,000 living people, but we were able to do it in 100 or in postmortem tissue from 100 or so postmortem samples, and found that indeed that C4A expression was elevated in the patients relative to the controls. It's not even clear this is the time in life at which these signals would be strongest. I mean, I guess ideally you might look in early adulthood or something like that, but even later in life, this signal was clear, and in fact, this difference is stronger than can be explained by the genetic results on its own. That is to say, it may suggest that other genetic influences also shape expression levels of C4A in the brain, or other environmental exposures, or something like that. So the genetics of the C4 locus only explain about a third of this difference between cases and controls. So, of course, this just took one mysterious question and replaced it with a different mysterious question, which is, if C4 is a driver of this association signal at the MHC locus, why? What is C4 doing in the brain? And we started out with just asking, well, where is C4 in the brain? And very quickly found that it was actually present at synapses, that C4 co-localized with pre- and post-synaptic markers. Now, this was interesting because so much else about the genetics of schizophrenia points toward dysfunction at synapses, and in particular, rare loss of function mutations in schizophrenia patients are far more likely to be at genes that encode synaptic proteins than they are to be at a random gene. So C4 was kind of present in the right place, but what was it actually doing there? So I think here we could easily have gotten stuck or gone down the wrong path were it not for work that Beth Stevens and Ben Barres had done over a recent period of time. And sort of the history of this is that, when we studied about complement proteins in college, we learned about that they were this component of the innate immune system that tags cells and debris for elimination. And in particular, C4 instructs the deposition of C3 onto debris and unhealthy host cells and infected host cells, things that would be necrotic cells, things that would just be really good to clean up. And that C3 then acts as this kind of molecular post-it note that invites the attention of phagocytic cells like macrophages that come in and do the cleaning up. But what are they doing in the nervous system? So it turns out, so Beth Stevens had discovered when she was in Ben Barres' lab that complement proteins have this night job in the nervous system where you suppose you're a neuron and you want to decommission a synapse or a dendritic spine. So one way you could do it is you could just try to take it apart like Lego by Lego. And that's sort of one model. But Beth showed that there's a fast way to do it that tends to happen at particular waves in development when a lot of synapses are being removed really quickly. And that is that the structure becomes decorated with C3, and that is in a signal for a different set of phagocytic cells, microglia, the phagocytic cells of the central nervous system, to then phagocytose them. And so this was a very interesting potential connection, because as I mentioned, the one kind of pathological known in schizophrenia was this finding of reduced numbers of dendritic spines in autopsy studies from Fortunately, Beth and also Mike Carroll, who's an amazing immunologist that has a really encyclopedic understanding of the complement pathway, are both sort of academic neighbors of ours. We're in different buildings but on the same campus, and we all immediately started having kind of a joint lab meeting this continued actually to this day to try to make sense of all of this. And in fact, the work that I'm going to tell you about was emerged from that. So synapse elimination is something that's been well studied in the mouse visual system, where there's this model developed by Carla Schatz and others of based on thalamic relay neurons that early in development have many synaptic inputs from both eyes. But then after a certain critical period in development have now reorganized into, they have many fewer inputs, and they've now reorganized them into eye-specific territories. And Alison Bialis, a postdoc in Mike Carroll's lab, who was previously a graduate student in Beth's lab, showed that C4 actually drives this deposition of C3 at synapses. That is to say that in mice that lack C4, you don't see nearly the same level of deposition of C3 at synapses. And that C4 is expressed by precisely the neurons whose synapses are being pruned in the system, and that C4 seems to regulate the extent of synaptic pruning. That is to say, in mice that lack C4 or that have fewer copies of C4, that far more of these neurons tend to retain multiple overlapping inputs. So that's all. More recent work by Jesse Presume and Alison Bialis in Mike Carroll's lab has actually been, they actually set about the brave experiment of trying to understand the differences between C4A and C4B but in an animal system, right? But the problem is that this functional specialization of C4 into C4A and C4B occurred in primates. So how could you study it in animals? So they have actually set about to introduce the human C4 alleles into mice on back transgenes, bacterial artificial chromosome transgenes, and also in a parallel set of experiments, now they're also doing it by CRISPR. So they created mice that lack mouse C4, but instead of have either human C4A, the one we found to be associated with schizophrenia, or human C4B. And as they expected, and this is just to control, it was known for a long time that human C4B, but not C4A, can drive hemolysis. And in fact, they showed that these introduced human alleles in mice are the exact same thing. So if the ones in which you've introduced human C4B, that have this strong complement-mediated hemolysis effect in this assay that C4A lacks. Actually, C4A was for a long time, because of assays like this, thought to be just kind of a less functional or a non-functional C4, which is one reason our genetic result pointing really specifically to C4A had been surprising. But Mike's lab has found that, for example, now that human C4A but not C4B restores synaptic pruning in mice that lack C4. And also, the human C4A, but not C4B, restores the efficient engulfment of synaptic material by microglia in mice that lack C4. So this is all work that's in progress, but that I just wanted to say that it does seem to be possible to explore in an animal system how these the differences between these human alleles, and that produces that seems to the results seem to align with the human genetic result in suggesting that there are functions in the nervous system that C4A has but C4B does not. So I'm going to make sure we have time for the next part of this. So and I think many of these things are kind of known to you guys. First of all, that many other genetic results in schizophrenia point toward diverse dysfunctions at synapses. It hasn't really triangulated on any specific pathway. It's just sort of many, many different kinds of genetic dysfunction at synapses seem to increase people's risk of schizophrenia. But certainly one idea that's worth thinking about is that that synaptic loss or pruning could be an integration point for many different kinds of synaptic dysfunction. That is to say, many different kinds of synaptic dysfunction might be upstream of the recognition by cells of underperforming synapses, and they're decommissioning via pathways like this. And it's quite possible that this process becomes particularly intense in the association regions of the cerebral cortex during adolescence and the early 20s, because that has been seen to be the period of dramatic reorganization of synaptic circuits and loss of dendritic spines. And there's actually just a wonderful and prescient piece that was written by Erwin Feinberg in 1982. He's at UC Davis now, but was actually once spent quite a bit of time here working with Rob Malenka and others. And it was, when Ashwin was writing up his thesis and we found this, we were just amazed at how much he had anticipated about these kinds of potential mechanisms just from kind of thinking very intuitively about what he was seeing in adolescent patients. It was long before molecular factors for any of these things were known, but he was already kind of sort of drawing at least potential connections among these processes. So this is what I've told you about so far. Is ways to study extreme forms of genome variation, that many of these alleles affect expression of C4A and seem to associate with schizophrenia in proportion to that effect, and that a potential mechanism of this, although we don't know for sure obviously if it is the mechanism, is that C4 seems to regulate synaptic pruning and may connect to this core pathological observation. So now I want to tell you about some more of the new work that I alluded to that I'm really just sort of discussing here today for the first time. So I apologize if some of it is a little bit rough. But I'm really eager to get the comments of people here about it. So one of the things that Mark Daly has long noticed about these genetic associations in schizophrenia is that there's a strong inverse relationship between odds ratio and allele frequency. That is to say, the alleles that are really common have a sort of a lower upper bound on their effect size than alleles that are sort of somewhat lower frequency in this 1 to 5% range. And this, as exome sequencing studies eventually discover even rarer alleles of larger effect, potentially this U-shaped plot will even go kind of like farther. It'll require a higher y-axis, but one might expect to continue seeing the same relationship. And this probably reflects that people with schizophrenia are generally greatly impaired on average in terms of fertility. They tend to have about a quarter as many offspring as people without schizophrenia, and that probably just reflects sort of historical selection against alleles of large effect. But it turns out that Mark actually made this plot before we had kind of worked this out about the MHC locus and the C4 alleles. And the C4 alleles somewhat stand out from this larger pattern. That is to say, given the risk level that they confer, which again this isn't like big B, little b genetics, this is still like just a quantitative risk factor, but given the level of risk that they confer, they're unusually common. And that really got us to wondering about why they might be so common. And ultimately, that led us in a way that I will tell you more about in a minute, to the genetics of lupus, of systemic lupus erythematosus. This is a systemic autoimmune disease in which the immune system attacks healthy tissue in many parts of the body. The usual onset is kind of 15 to 45 years of age, although there can be older onset cases. It's typically treated with steroids and other immune suppressants. The cause is unknown. And something else that I'm going to get back to in a minute is that there's a strong age bias in the incidence of it. So women of childbearing age are affected about 9 times more often than men are. So actually, one of the real thought leaders in the genetics of lupus is actually UCSF's own Lindsay Criswell, who I think is actually not here today, but Lindsay has contributed to or led so many of the results on this slide, together with many of our other colleagues in the lupus genetics field. So the strongest genetic association, as in schizophrenia, the strongest association in lupus is to SNPs in the major histocompatibility complex locus. The association is actually much stronger in lupus than it is in schizophrenia, even though the sample size actually isn't as large. And it has kind of long been thought that this is due to variation in HLA genes. And that's just a natural thing to assume about an autoimmune disease, in particular because in other autoimmune diseases and autoinflammatory diseases such as rheumatoid arthritis and multiple sclerosis and type 1 diabetes, there are HLA genes with extremely strong effects, and they turn out to affect the peptide binding groove of HLA proteins and determine which antigens can be presented. And in fact, in many cases, these alleles, like in type 1 diabetes, the risk allele is strictly required. Like if you don't inherit that allele, you have almost no chance of getting type 1 diabetes because it presents the antigen that's the culpable antigen in type 1 diabetes. Similar results in these other diseases. But in lupus, the genetics has kind of never fit that model as well. Okay, so, and Lindsay has done some really nice work on this, for example, showing that alleles in the MHC, they're risk factors for having autoantibodies generally, but there's not this connection of specific alleles with specific autoantibodies, or let alone with like specific changes in the peptide binding groove or anything like that. And like in schizophrenia, the observed genetic signal is complex, and it requires many different HLA alleles to explain. And yet again, there's no correspondence of those HLA alleles with the different kinds of autoantigens that people can develop during lupus. So we became interested in the question of, could the HLA alleles actually, as in schizophrenia, be genetic markers for something else? And this was as work that's been led by Nolan Kamataki, a student in my lab. So it turns out that what we had done in schizophrenia was actually much harder to do in lupus for reasons that'll become clear in a minute. And so we needed to create a much larger imputation reference panel. That is to say, we needed to know about C4 alleles and SNPs in a far larger number of people. So fortunately, we were able to take advantage of emerging whole genome sequence data from large cohorts. And so we made a new reference panel of C4 alleles and SNPs, but now in a genetically diverse population that included African Americans and Latinos and other populations, and 10 times as many people as we had been able to use before. So again, although this is the same figure as before, now we made a 10 times larger reference panel, and we're going to use those to infer what C4 alleles patients for various autoimmune illnesses have. And I'm going to represent the data slightly differently. So now for each person, you can imagine we take their SNP data, and from their SNP data, we decide that they have for example, this person, we figure out their two HLA alleles, and then we're quite certain they have this combination of C4A genes and C4B genes, and so that's represented here. Sometimes we're a little bit less, sometimes there's a little bit of uncertainty in the imputation, so they're represented here. Every once in a while, you get someone who has a recombinant haplotype that's not represented in the reference panel, so there's much more uncertainty about their state, but they're only 1 or 2% of the cohort. But basically, this lets us kind of assign people probabilistically to each of these different genotype groups as having different combinations of C4A and C4B gene copy number, and then we can ask, well, what is the risk of lupus in each of these groups? And the result was quite striking. So first of all, now here the area of the circle is just proportional to how many people have that genotype, and the color of the circle is relative risk for lupus. And we found that across these C4 genotype groups that relative risk varies almost 7-fold. That is to say, people in this group have almost 7 times as much risk as people in this group. But there are a couple of other patterns here too I want you to notice. So as you go from left to right on the plot, that's the effect of increasing C4A gene copy number, and you can see that as you go from left to right on the plot, no matter how high no matter what your C4B copy number is, the effect of going from left to right is a profound reduction in risk. Also, the effect of going from bottom to top, that is of increasing C4B gene copy number, is a modest reduction in risk. And then, which actually suggests that C4B might be protective also, but perhaps less protective than C4A. And the other way you can see that is if you just go diagonally down the plot, which is the effect of exchanging a C4A for C4B, then that's also a reduction in risk. So in other words, giving up a copy of C4B in exchange for a copy of C4A is a good deal. It reduces your risk of lupus. And I won't go through it blow by blow, but we did many of the other kinds of analyses that we did in schizophrenia to really establish that it was the variation at C4 rather than this other nearby variation that was driving this signal, and concluded that it did. And actually we're able to do some additional things here in terms of using genetic data from other populations in which the linkage disequilibrium is much less, and you can kind of break these ties. And it's really C4 that stands out as the consistent signal in multiple populations. So, but we also found that C4 appears to protect against another autoimmune disease of the central nervous system, which is neuromyelitis optica, in which people develop antibodies to aquaporin-4 and astrocyte protein. And again here, it's lacking C4A is a profound risk factor where people have 4 to 5 times the risk of people who have the most common C4 allele. So this sort of led us to an interesting situation, which is, it kind of brings back to this question asked earlier about how could these alleles have become so common despite having conferring the schizophrenia risk that they do. And in part of the answer may relate to that these same C4 alleles seem to have opposite effects on the risk of multiple autoimmune diseases of the CNS. And in particular, the same alleles that increase risk for schizophrenia seem to protect from lupus and neuromyelitis optica. And in fact, their effect in autoimmune disease, it's a far larger effect size than their effect in schizophrenia. That is to say, their potential for obviously it's hard to keep score of such things, but it would be hard to argue that they're a net negative for fertility in the human past, especially before the era of immunosuppressive medication and sort of successful treatments for lupus. So the same alleles seem to have opposite effects on these illnesses. But another very striking thing about both of these illnesses is that they affect males and females with substantially different burdens. And you guys are obviously quite familiar with this in schizophrenia, although it would be interesting to discuss it more. But there's, the incidence of schizophrenia in males appears to be higher than it is in females. It's about 50% higher. But this difference actually becomes more striking if you look at young adult onset cases. So cases that emerge kind of around the end of adolescence and early adulthood, it's more like a 2-to-1 difference, and it's somewhat compensated for that. There's this second wave of female cases around menopause and the 40s. Males also tend to have more severe negative symptoms and less remission and higher relapse. So this is a sign that they're kind of more severely affected. In lupus and NMO, it's very much the opposite. There's a much higher incidence in females. The female-to-male incidence ratio is about 9-to-1 among cases that arise during their reproductive years. Although, as with schizophrenia, there's less sex bias among cases that emerge outside of that window. So among cases that emerge much later in life or among children, it's much more similar between males and females. So. And this gets back to, I think, just one of the most fundamentally interesting questions really about all illness, which is why do males and females have different biological vulnerabilities? Let's say we get the same illnesses, but we clearly don't get them at the same rates. And this must suggest but what aspects of human biology can help us understand this? And Stefan has written many very kind of thoughtful things about this, and one piece I often point people in the lab too is actually this piece that he wrote for the Simons Foundation about the female protective effect in autism, which is also extremely, it's actually much stronger in autism than it is in schizophrenia. So C4 seems to show these opposite effects in these illnesses. They also have opposite sex biases. So a natural question then suddenly became, does the effect of C4 alleles differ between males and females? I say, if we look at males and females separately, is C4 more important in one than the other? The answer turns out to be yes. And this turns out to be true in both lupus and schizophrenia. So that is to say, if we look across this entire allelic series, each allele matters in both males and females, but it matters more in males. That is to say, it spreads the relative risks. The allelic differences spreads the relative risks more strongly in males than it does in females. Probably even more so in schizophrenia, actually, than it does in lupus. In schizophrenia, it's almost a 2-to-1 ratio in the effect sizes of the C4 alleles. So again, the genetic variation seems to matter in both groups, but to matter more in males. And this is not true of other genetic effects in these illnesses. People have looked very carefully at this, and almost every genetic effect that's been mapped in both illnesses shows the same effect size in males and females. That is to say, very little else about the genetics has ever suggested that the biology of, pointed towards something different about the biology of the illnesses. And also, there's never been really been, there was one point in this idea that this might be due to like X-linked genes or something like that, but that's been fairly exclusively excluded in all of these illnesses. So, but this is a genetic effect that seems really different between males and females. So why might C4 alleles shape risk more strongly in males? So here's just some ideas. So if the C4 genes are just expressed more highly at an RNA level in males, then the effect of allelic differences would be magnified, right? Because it might just kind of act multiplicatively with a stronger baseline tendency to be expressed. Or the effect might be at a protein level. It might be in the processing and secretion of C4 proteins or some regulation of their degradation and half-life. Or there might be differences in downstream responses to complement. So the first one turned out to be very easy to rule out because with the data from this large gene and tissue expression consortium, we could just really quickly go and look at male-female differences in every tissue, and there was no substantial differences in expression of C4, or indeed in the other components of the classical, because you can imagine the male-female difference might not have to be at C4, it might be in some other component of the pathway, but when we looked at all the components of the pathway, they're expressed at the same level in males and females. But it's a very different story when we started to look at protein. And this took us longer because cerebrospinal fluid, which is really the kind of relevant bodily fluid when you're talking about the central nervous system, isn't collected as easily or as casually or as frequently as plasma is. But as we accumulated enough samples, the following pattern became quite clear. And now we've now seen this across multiple CSF panels from different people, different parts of the world, which is that in cerebrospinal fluid, males tend to have higher concentrations of C4 and C3, its effector, than females do. And these plots understated a little bit because this is on a log 10 scale. It's about a 40% increase in males relative to females. And of course, we don't know what the underlying math of a lot of these things is. It may be that what matters isn't the shifting in the mean. Maybe it's just how often you're in the far tail. But males are also greatly enriched in the tail of people that we see with very high levels of C3 and C4. Across individuals, although C4 and C3 are encoded in different genes on different chromosomes, and in many ways we've found from our single cell data produced in different cell types, across individuals C4 and C3 protein levels in CSF are also strongly correlated with each other. That is to say, the same people tend to be high in both. This may suggest shared influences on their production or secretion or degradation. But the combined biological effects of concentrations of multiple pathway components could in principle be substantial. And I just kind of want to leave you with the thought that this might contribute to sex-specific protective effects in these illnesses. That is to say, this might be part of why males seem to be somewhat protective from these classes of autoimmune diseases, but females seem to be protected from schizophrenia. In a relative sense, obviously. So just to sum up, alleles of C4 that contribute to schizophrenia risk also appear to be protective for multiple autoimmune diseases. These alleles, each of the alleles seems to affect risk more strongly in males than in females. And finally, these genetic effects may act multiplicatively with underlying sex differences in ambient levels of C4 and its effector, C3. So, there's still so much more that we need to learn about C4, and I actually just want to leave you with a couple of the questions because I don't want to sound. Sometimes I feel like some of the conversation about results, and particularly the way like when things get picked up in the newspaper and things like this from our earlier paper can make sort of things sound more certain than they really are. And so I just want to clarify that there are some things that there's sometimes an ambient conversation about is established by our results that I don't think is really established yet, and I want to emphasize that the things on this slide are really still questions. Okay, so one is, are the primary effects of complement on schizophrenia risk, are they on the initial pathology, or are they on the progression of the illness, this kind of worsening downward spiral? We really don't know, okay? So it's true that the genetic studies are on whether you have schizophrenia or not, and it's tempting from that to conclude that what you find are risk factors for getting schizophrenia, but it's always strongly confounded by ascertainment, right? So, and in particular, the more sick you are, the more frequently you're going to the hospital, the more often you're seeing doctors, the more likely you are to get ascertained into a genetic study. And in fact, some of the differences that we see in the strength of these genetic signals, cohort by cohort, seem roughly to go along with that. That is to say, we see stronger signals where people, for example, ascertained treatment-resistant patients that didn't respond well to antipsychotics, or where they ascertained people who had many hospitalizations rather than ascertaining in outpatient clinics, for example. So I would say this is just something we still really don't know, and so I want to emphasize that. Sometimes there's conversation about, like, should we be like trying to prevent illness with complement-based therapeutics? And I just think it's way too early to be talking about that. So, and also I think, if anything, our results about these sex differences, and again, here's where I'm going to be really interested to hear your thoughts. To me, what's more striking about the male-female differences is less in incidence than it actually is in severity. But I would love to hear the thoughts of people who have interacted with a lot more patients than I've had the chance to do, not having had that kind of training. So then the second question I want to also leave you with is synaptic pruning or another biological process really the mechanism? So, obviously, in this work, we felt like it's important to make the connection between, to see whether it really is true because C4 wasn't itself known to instruct synaptic pruning. Pruning had been associated in mice with other complement components. We thought it was important to make that connection and to show that, for example, in mice, these alleles do instruct synaptic pruning and things like that. It doesn't necessarily follow, though, that that is the mechanism by which the genetic variation is increasing risk. It's very plausible because it connects to this pathological known, but I feel like over and over again in science we make the mistake of that we're also eager to connect the dots, that we connect the dots that we see that look close together, but often with time, as more dots come out on the plot, there turn out to be other paths too. And a lot of these protein, for decades, what we knew about the complement proteins had to do with their role in the innate immune system, right? And then Beth and Ben discovered the second thing that they do. This is a very versatile molecular system. You could potentially use it to do lots of other things. You could use it you could clog up ion channels with complement protein. There's potentially a lot of things it could do. And we as in science, we kind of blindly find our way through these things one at a time and always assume that the latest one is going to be the answer. And I just want to underscore that we don't know yet. There's still a lot more work to do. We should certainly keep thinking about it and further develop the hypothesis, but we should also stay open to other things. And so I just want to leave you with that and thank the many people that contributed to this work, both in my lab and most of our colleagues at HMS, but also the genetics consortia that generated a lot of the underlying data that we were able to reanalyze here. And I know people here have contributed to the Psychiatric Genetics Consortium, and also that Lindsay and others have been leaders of the Lupus Genetics Consortium. I just want to thank everyone for their contributions to all of those activities along the way. Thank you.
Thank you for a fantastic talk. We have some good time for questions and our esteemed leader.
Steve, thank you. Just such a fantastic talk. A triumphant return. I really like it here. One thing I want to say is I just want to thank you so much both for people talking, also for the last slide. I think that it's a beautiful way to end, and I just want to, it's our major every molecule we're finding is biologically pleiotropic and does a variety of things, and depending on when and where you look, and you can find different characteristics. So I'm wondering, so it's a long question.
Yeah.
For us,
We have half an hour, so yeah.
When we find rare variants, we have Stefan's favorite gene, SCN2A, 30, 40, 50-fold increases in risk. And so we have these huge main effects, and we still have no idea when we look whether or not we're looking at something that is really reflecting the pathological process. And with common variants, I think you have clearly the same problem, because you see something doesn't necessarily mean that it. But I think, in my view, you also have this additional question, which I think is answerable, which is your effects are much, much smaller for any individual allele.
Yeah.
And so how do you think about sort of what's the path forward for you to end this talk by saying, we think that there's a really strong body of evidence that suggests that this is a mechanism that's been elaborated by the genetics, and this is something that we ought to be pursuing with regard to thinking about therapeutics or prevention, blah, blah, blah. How do you get from where we are now to where we want to?
Absolutely. So, and actually, I think, so first of all, I just completely agree with you. We know far less about the biology of these things than we think we do. Because a gene has a name and some annotated function, and stuff comes up in PubMed when you PubMed it, we think we know what the function is. And almost every turns out to have many functions. The same molecular system gets reused to do lots of different things, because it's far easier for evolution to find new uses for parts than to just kind of invent a new part from scratch, right? And so, and in general, a lot of these molecules get named for their function in the immune system just because the immunologists figured that out first, right? And then we learn a second thing about it, and then we think that must be the answer. But we don't know. We don't know. I completely agree with you, and I also agree with you that that's just as true about. For that question, it doesn't matter whether it was implicated by a rare variant or by a common variant. We just really don't know nearly enough about the function of these genes. It was one reason we put so much work into developing DropSeq, is because we didn't even know where most of the genes were expressed, right? And we wanted to kind of create a world where you could just kind of look those things up quickly in a data resource and compute on it and things like that. But even then, that only gets you part way, because a lot of these genes have very broad patterns of expression, right? And so then you really get to the question of where does the expression of it matter, right? So what's the effect of not having, and which cell types care about not having it, right? So there's potentially a couple paths into that question. So I think with rare variants, where that lack might transform the biology of a specific cell, with SMN and motor neurons and in SMA, where you might hope with single cell analysis of brain tissue from patients that you could really say, either this cell population disappeared, or so there was some cell death among it, or this cell population is still here, but it has a kind of transformed gene expression signature. And I really hope that will contribute to unraveling some of the mechanism of these rare variants, but of course that's going to really take, and one, I didn't have time to talk about it today, but one of the areas we've been able to make a lot of progress with DropSeq this year is to where we can now analyze nuclear transcriptomes and thereby generate actually really good data on fresh frozen tissue from brain banks. And one of the reasons I feel like that's going to hopefully be really important is there you can really start to get at cases and controls. And a lot of these brain banks are big enough that now if you go back and start genotyping or sequencing the genomes of people in these brain banks, you'll find some of these mutations, especially in autism, right? And then you can start to see, which cell types, granted, SCN2A or something is really broadly expressed, but which cell types really, were different when they didn't have it, right? Or when they had only one copy of it. So with common variations, a little bit different of a problem, because I don't think that these common variants are grossly distorting the biology of any one cell type. These are more like genetic nudges. And so what might be a path forward there to get into the kinds of insights we'd all like to make about what are the cell populations and therefore the circuits that might be involved in each other. So what we're starting to see, at least with the common variation, is that even though the gene is broadly expressed, that the specific allele or haplotype often affects only one specific part of that gene's expression. Okay, so that is to say, a common variant in, for example, common variants in 4 different subunits of the L-type calcium channel associate with risk of schizophrenia. They're all very broadly expressed in many, many neuronal types, but a specific allele is often causing a quantitative change in expression level in just one or a few sister subtypes or something like that. So that might be a path toward trying to use the common variation to triangulate on the cell populations that matter. So I think the paths forward from both. So the strength of the common variation might be that the genetic lesion itself is cell type specific in where it acts. With rare variants, it might be that the consequence, that there's a more dramatic consequence in a specific cell type. But that either way, the single cell analyses of brain tissue, you might be able to get to the bottom of it. And one reason I didn't talk about that is we're really excited of having kind of further developed dropseq to be able to do that, but we're still so early in generating the data. And in fact, we wanted to do it in lots of samples with schizophrenia, but we actually just decided to start with experiments in neurodegenerative diseases because they were pathological knowns, and we could just ask, do you see the stuff that you'd feel dumb if you didn't see, like loss of SPMs in Huntington's, for example. And so you totally see those things. And you actually do some of the patterns of microglial activation in these neurodegenerative diseases are actually surprising and interesting because they're so unlike the in vitro models that you realize that a lot of the in vitro work on the glial activation is just not really. It just doesn't look at all like what you're seeing in vivo. You see transformed glia, but they're not transformed in ways we probably have a good in vitro model for today. So that's encouraging enough. That's enough that I think we are going to start trying to do this in schizophrenia. But that work's really just starting, and so I just have much less interesting to say about it, other than that I'm kind of hopeful about it. But then the third thing is that the sensitivity to a genetic lesion, whether it's a common variant or a rare variant, might not just be a function of cell type, or might not be a function of cell type at all. It might be a function of condition, right? It might be that something becomes important to a neuron. Is it important to a neuron not all the time, but right after it's been active, or when it's in some kind of synaptic remodeling mode, like it might go through during adolescence, or when it's just been exposed to cytokines or something like that, right? And we're not probably going to find condition-specific genetic relationships by just looking at kind of endpoint postmortem tissue. That really is going to require experiments on living cells. And so Kevin Egan and I have been trying to develop experimental systems, we call them massively mosaic experimental systems, where we mix cells from hundreds of different people and then assay phenotypes in them, and then use single cell analysis to assign each cell's phenotype to each individual person. So we could say for each person, that this person's cells had this distribution for this phenotype, whereas this person's cells had this distribution for this phenotype, and start to do sort of like quick genetic association studies in a dish, but in a single reaction vessel, and there the hope is not just to look at common variants, but to look at rare variants too, and especially to look at what I feel like is one of the really compelling questions about rare variants, which is why, especially in schizophrenia, which is why is their effect so strongly background mediated? So if we could look at the effect of a loss of function mutation on 20 different genetic backgrounds, I think that's something that we would really like to see, because even where we are getting hints of rare variant effects in schizophrenia, it's clear that the penetrances are much lower than they are in autism. And even like the 22q C and V, it's only about 25% penetrance. So it really, you have to kind of study it on multiple backgrounds. So we're sort of developing these population in the dish experimental systems so you can try to get it, looking at how genetics shape phenotypes that you can only measure in living cells that are reacting to things and undergoing dynamic responses and things like that. So that work's also fairly early, but it's one of the things I'm most excited about going forward. Yeah.
So, Steve, we've known for decades And there's even some evidence that there is a faster loss of gray matter over time in some longitudinal studies than we see with normal aging. We also have had the postmortem data for some time that show the reduction in neuropil and the reduction in synaptic spine density. But it is far from accepted conclusion that these postmortem changes are the molecular correlate of the gray matter reductions we see with MRI.
That was what I just wanted. I almost interrupted your question so that I could ask you that, because I realize people make that connection in meetings a lot, but it seems a little bit facile. And so I just wondered what is
One could look specifically at some of the work and writings of Danny Weinberger as a very vocal skeptic of the association between the gray matter findings and these postmortem findings. But it brings me to a question about the C4 story, which is that it seems to me there are a lot of bridges that we have to forge between that finding and the human data. For one thing, starting with Feinberg's hypothesis, the idea there was why does schizophrenia, or what part of what he was trying to account for was why does schizophrenia tend to emerge in late adolescence or adulthood? And at that time, we were just starting to see the gray matter loss at that time. And based on some, actually what, looking back, seems like pretty limited literature out there on adolescent pruning and evidence for it. Some of it comes from Peter Gutenlocher's postmortem work in humans. But there's actually, other than that, fairly sparse animal data to support adolescence as being a time of accelerated pruning. And having had this conversation with Beth Stevens, where she really thinks about her work and what she's documented in connection with the complement proteins as subserving early brain development, and not in her models something that we would associate with adolescent maturation. So trying to think about how we bridge the C4A effects to, number one, adolescent brain maturation and pruning, if it exists in animal models. And number two, is there a way to take advantage of the MRI data that are available from some probably substantial subsets of the individuals who've contributed genes to the sample that you studied to ask basic questions like, does C4 allelic variation correlate with gray matter changes or gray matter volume in even a healthy adolescent sample? That is, can we try to pull out some signals that would relate the C4 allelic variation and the pruning it presumably regulates to changes in what we see in vivo with MRI?
So many good questions there. Let me just start with one about sort of what do we really know about adolescent cortical maturation? And the only answer is not really. Very little and not nearly enough. So it is clear that different parts of the cerebral cortex, I think, mature at different times. Okay, and that primary sensory cortex tends to mature and prune and myelinate very early in development, and these parts of the cerebral cortex, this kind of distributed association cortex, it's expanded so much in human evolution, and it's really much more connected to mentation and thinking about our problems and planning and executive function and things like that. It's greatly expanded in recent primate evolution, and it also, lots of things point to that it matures much later, right? How much of that is pruning versus myelination versus, really not as well worked out as it needs to be, but I think lots of things point to that's a late maturing part of the cerebral cortex, in which the maturation process actually continues into the third decade of life. And to me, it is a greatly understudied aspect of developmental biology. Developmental biology tends to focus on things where there's sort of more dramatic epochs of cell birth and cell death and cell migration and things you can see under a microscope. And so I think result, although cortical maturation although I still think it's fundamentally a developmental process, it's developmentally timed, it's happening in people who are legally adults, but we still need to think of it as a developmental process and to try to understand it in a developmental context, but that may require different kind of tools. So that's another kind of thing we're really hoping to do with single cell analysis, is where a lot of surgical resections for epilepsy are in young people, in the teens and 20s. And so we're really trying to sort of recreate just even a natural history of how the different cell populations are changing from age 10 to age 30. And what's changing at what time, and what events are early, and what events are late, and because I just think we still really don't know about these things. Now, the reason Beth has focused on the visual system in her earlier work, she's not exclusively focused on that now, but the reason Beth in that field focused on the visual system isn't that they don't think these things happen in other parts of the brain, but you can do rigorous experiments in the visual system because you can control the inputs. So you have systems where you can do these experiences around you close the left eye, close the right eye, and inject things in the left eye and the right eye and things. And so you can just do better experiments than you can do in other parts of the brain. But also rodents don't necessarily have a great model for association cortex. And association cortex is clearly something that's just changed a lot in primate evolution. And so it's not totally clear to us that you can study this all that well in primates. And in fact, that's one reason we've been working with closely with Will Pink's lab to try to develop marmoset as a model system. And one of the things we're already seeing in the drop-seq experiments is there are tons of things that marmosets have in common with humans in terms of cell composition and the cerebral cortex that neither of them shares with rodents. And so again, it's just another area where I think the only fair thing to say is we really don't know. It's going to take a lot more work. Yeah.
Have you looked at age of onset and the C4 expression data in schizophrenia and in line with previous comment? It's been, I don't think it's necessarily either or in terms of the early childhood versus adolescent onset. I mean, it seems to me that there's evidence that this is, could be progressive on a two-hit hypothesis. But I mean, there's evidence that even individuals that are first diagnosed in late adolescence can be recognized as unusual, at age 4 and 5 in movies and things like that.
Yeah, it's a really good idea, and we've just not enough time in the day, and we just have not gotten it. Yeah. I think it's a really interesting thing to do, and we just have never had time to do it. Matt?
I wonder, I may have missed it, but I would love to hear the answer to the second half of the question, which was, do you think that doing imaging genomic study in a healthy population or a disease population or both, looking at a single allele like C4 and asking the question of whether or not you should see correlations with gray matter varying sufficiently in the population? I just, I think it's a question that comes up repeatedly, and I'd love to hear your thoughts on it.
Just to put a finer point on it, there are some longitudinal MRI data sets, and in particular, some that are being collected even now in children and adolescents. And the question is, would people who have more predicted C4A expression be expected to show steeper declines in gray matter during adolescence?
I don't want to discourage people from doing what they want to do, but I just want to maybe bring up the reason why it might not work. I think, for common variation, C4 is the largest risk factor that we know of. But again, we're still not in the domain of big B, little b genetics here. It's just a quantitative risk factor. I like to think of it as that it kind of moves a threshold, right? But lots of other things determine whether that threshold is crossed or not, and other things probably move that threshold too. And so I think that what's exciting about C4 is just we know the alleles and at least the first order things that they're doing, but it doesn't follow from that it's going to be this hugely predictive thing. it's still a fundamentally polygenic illness. And so I think that's the reason why suddenly looking at C4 with like all these other phenotypes might not work. But I don't want to discourage people from trying any good idea. But that might be the reason I have like sort of research. And it's also, I feel like we don't know, but I feel like with time I tend to feel more and more like it's possible that the C4 and complement part is more affecting the progression of an illness after some other pathophysiology has already started. Not that couldn't still be incredibly important if you could slow it down or stop it. I mean, sometimes I think we fetishize cures when a lot of, if you could, even if you can't make something whole, if you could stop it from getting worse, you could still, that could be very important in terms of health and impact. But I think there's many reasons to start to think more and more that this biology might be more related to the biology of progression. And another is that just, at least in the common variation side, it's estimated that about 2/3 of the genetic influences on schizophrenia and bipolar disorder are shared. C4 and MHC is very clearly something that's not shared. It's very clearly something that's specific to schizophrenia. And so, maybe it's useful to think of it as there's some earlier brewing pathology that's common to both disorders, and then in some people it can go down this particular path, and C4 may be shaping more whether it goes down that path, or this pathway may be shaping more whether it goes down that path or not, but still not be like the founding, the sort of founding molecular, or the sort of founding pathology. So that's kind of another reason why.
Do you think it's as I agree that disease is complicated to try to account for with this one gene and its effects, but what about just something like the observation that gray matter does decline starting around age 10 in human MRI studies? Is it also sort of futile in your mind or unlikely to work to look for C4 gene variation as an explanation for variation in gray matter decline in healthy adolescents?
I never want to discourage a good idea, but I think it can be, and we've actually helped a lot of groups sort of impute C4 into their SNP data and look at some of these things. But again, the reason it might not work is that I think there's a substantial chance that that's a complex phenotype that's shaped by very rare and common variation in many sites of the genome, and that it would be hard to pin it on any one thing, or even conclusively implicate any one thing unless the sample size is very, very large.
Good, thank you ever so much. We are at time. Thank you for the wonderful questions and, of course, the excellent talk. Thank you.